Hi, everyone. It's a pleasure to be speaking with you all today. Again, my name is Alex Olshansky. I'll be presenting this work on behalf of my colleagues, Cassandra Hayes, Chaitra Kulkarni, and Kerk. We come from the College of Media and Communication at Texas Tech University, and this project was funded by an NSF career grant. Again, the title of our talk today is The Characteristics of Influencers and Opinion Leaders in Science Gateways and Cyberinfrastructure. And so when we refer to these influencers, what we're talking about is this concept of opinion leaders, specifically Everett Rogers' diffusion of innovations theory concept of opinion leaders. And so Rogers was a widely cited communication scholar who developed this theory of diffusion of innovations theory that describes how new ideas, new technologies are adopted and spread through social systems over time, and what factors lead people to either adopt or reject new technologies. And so Rogers described these opinion leaders as these individuals who have this kind of informal influence over others' attitudes and behaviors. They're typically respected by a large number of their peers. They're socially well-connected. They have a network of followers. This is kind of what makes them opinion leaders, is they have this group of people that kind of follow their lead, follow their opinions, and they're typically early adopters of new technology. And so why these CI influencers are important is as science gateways and CI advances further, outreach becomes increasingly necessary to sustain its growth. And so these influencers and opinion leaders play a fundamental role in accelerating the adoption and diffusion or spread of technologies like science gateways and CI. And so here we compiled insights from a variety of CI stakeholders to offer a systematic description of science gateways and CI influencers, who they are, what qualities they possess, how they communicate, and why they are successful at influencing others. And so to do this, we analyzed transcripts from 132 interviews with CI stakeholders who were interviewed between 2016 and 2019. And these included domain scientists, computational technologists, supercomputing center administrators, among others. And at some point in the interview, participants were asked how they were first introduced to CI, and if there were any particular people that influenced their involvement. And if they identified specific people, they were asked a follow-up question about what made these people influential or convincing. And so our team conducted a systematic thematic analysis of these transcripts where 3 team members used a software called In Vivo that allowed us to tag certain chunks of text within these transcripts that contain certain themes. And here we were focusing on this influencer theme when participants were describing their influencers and what made them influential. So once we tagged these chunks of text for these themes, we had another 2 team members then analyze these chunks of text for common sub-themes. So our first research question asked, who are these CI influencers? And so we find that influencers exist across different roles. They can be CI users, they can be non-users, CI developers or outreach educators, but essentially they're influencers because they're introducers. They're the ones who introduce CI to others and pass along this essential knowledge. We also find that CI influencers facilitate growth with CI at 2 fundamental levels. So first they play a mentoring role. So they're typically people like professors, advisors, or supervisors, et cetera, that help individuals grow. But they're also active and visible in their particular academic or professional communities, helping their communities grow. So here we emphasize this growth with CI at 2 levels, at the individual level, but also at the community level. Our second research question asked, what qualities do these CI influencers possess? What makes them influential? And so analysis of our transcript suggests that these CI influencers are boundary spanners. So these are individuals who seek out and bring new ideas into their community. These are people who reach across the boundaries of their communities and are able to bring in new information. And so some of the qualities that the participants described about their influencers were things like being visionaries or innovative and cross-disciplinary. CI influencers also possess these kind of reputational qualities. So when Rogers described opinion leaders, he said that they are effectively opinion leaders because because of their reputations, right? So he described examples like village elders or religious leaders. These are people who have certain types of reputations that allow them to influence others. And so the qualities, some of the qualities that participants described about their influencers were things like being respected, trusted. They were high flyers in their communities, typically regarded as movers and shakers, people who could get things done and move the community forward. Our third research question asked, how do these CI influencers communicate? And so we broke down our analysis into the what, why, and how of CI communication. So we find that CI influencers are really good at explaining what CI is. They're really good at articulating why CI is important for science. And they're really good at communicating this with excitement and passion for CI. So these are the kind of three fundamental levels of communication. And so to wrap up, we want to leave you with some tips and guidance for CI communication and influencer development based on the findings from this study. So if you are in a position where you are communicating about science gateways or cyber infrastructure, these are some tips for effective message design. So we suggest that message design that is to promote CI diffusion or spread should include what we call these 3 E's of communication. It should effectively explain what CI is, educate about why CI is important for science, and do it with this kind of excitement and passion. CI messengers will be most effective if they possess these mentoring roles that we described above. And again, these can come from users or non-users, developers, outreach educators. But these mentors should be facilitating growth with CI at these 2 fundamental levels, the individual and the community level. CI messengers should also possess these influencer qualities that we described earlier. So boundary spanning qualities and reputational qualities. And so effective science gateways and diffusion strategy should target opinion leaders within the science gateways and cyber infrastructure community and partner with them to promote SGCI. Users who are growth eager and growth hungry will respond better to influencers' persuasion. Otherwise, these persuasion efforts may be wasted. As right now, we see many efforts are this kind of spray and pray approach. Not that that's necessarily a bad thing, but as human efforts and time are limited, this kind of spray and pray approach can be rather costly when a more targeted approach may be more effective. And so with that, I thank you all for your attention and I open it up to questions and discussion.
Any questions?
Yes, if I may ask a question. Appreciate great presentation. I enjoyed it very much. I would love to hear your comments directly addressing what we're struggling with in modern day America, culture, and society with regard to the disinformation, misinformation, malinformation wars. How do we counter the disinformation, misinformation, malinformation when trying to promote a new approach or a new system or a new solution to a problem?
Sure, that's a good question. Yeah, and misinformation is a big deal these days. I would suggest that one of the best approaches is fighting bad information with good information. And so the best thing to do is to kind of get more of good information out. But I would suggest that if you're communicating about such complex topics like science gateways or cyber infrastructure, to communicate in this kind of approach where you also do it with these 3 E's, right? Explain what and why CI is important, but also do it with passion. But also a good way to help communicate these kinds of complex topics is using metaphors. So if you use metaphors like, I can't think of one, right? It's like infrastructure, like city infrastructure. Or if you're talking about the big data, right, you can use things like the depth of the ocean or things like that. These help people understand the concepts a little bit better, particularly if you can use metaphors that those people relate to and they can understand.
If
Okay.
I can make a quick comment as a co-author, I think that's why you want to work with opinion leaders, because opinion leaders and influencers are the trusted individuals with existing relationships, and that can combat the impression of misinformation and disinformation. Thank you.
Okay, so in the interest of time, we're going to move on. If you could stop sharing, I'll let the next speaker, Bailey, take over. Thank you. Bailey, please share your screen and start.
Yes, of course. I'll share my screen right away. I was looking at the old schedule and thought I was booked for 1:45, so I'll start it right now. Share my screen. It says you've disabled screen sharing, I'm afraid.
You should be able to share.
The button is telling me otherwise, I'm afraid.
Okay. Can you try again?
Yep. There we go. Perfect. Thank you so much.
Okay, sorry.
No worries. Here we go. Perfect. So, hello, everyone. My name is Bailey Mann. Today, I'm going to be speaking on behalf of my fellow students, Kaushik Ganapathy, who is a PhD student at Scripps, and Jiaxi or Jenny Lei, who is also a graduate of UCSD. Today, we're going to be talking to you about a model of GeoAct, or Geographically Assisted Agent-Based Model of COVID transmission. We've developed a gateway for use by schools to help determine the risk of various different intervention methods. And so here we go. To start the presentation, what's the problem? The problem that we primarily face here is trying to keep students and teachers healthy in the times of COVID-19, when different intervention methods seem to have different results, and that there aren't particularly the surety that we have in our protection of our children is not particularly significantly thought through. And so to help schools and teachers and parents decide what protections are safe and what protections are justified to help get kids both A, back in school, and B, avoid having them be at greater risk than they need to be, we need to provide them with the ability to look at the end results of these complex models that people can produce without needing them to be able to understand every step of the way. And so in order to kind of de-obfuscate a lot of the complications that go into determining how many people might get infected in different cases, we need to develop some sort of system to help the schools prepare by simulating whatever plans and intervention methods they might have. So we have a quick simulation video here, of which I'll show you about 10 seconds. So using this agent-based model, we simulate students, which you can see in the 3 different colors here of green, yellow, and red, moving around individually, and their movement is simulated randomly. The distance that they are away from other students is simulated randomly, and the justification for our model is based off of both aerosol and droplet transmission, which occurs within enclosed spaces and across different distances. So the model is, as you can see, particularly computationally intensive. And so the video here is a shorthand to give you an example of what it looks like. To move to the next slide, hopefully. So our initial solution was to have an SEIR-based approach, or susceptible, exposed, infectious, or removed, and to use the transitions between these different states for each agent in our model to determine at the end the effectiveness of the different intervention methods by comparing the rate of infection over time, over the course of weeks or months, given different initial conditions. The shortcomings, however, in this method, as relatively obvious from the previous slide, apologies while I move the Zoom controls out of the way. The shortcomings were an excessive runtime, which was caused by inefficient looping through the stages and the time steps to try and compute the infection at every single step. Limited access to model because it required use of the command line and knowledge of the specific inputs to set up the initial conditions. And as a result of that, we had to present mostly pre-computed scenarios, such as the previous video for the web-based version. And also as a result of this, we had limited access to NPIs or non-pharmaceutical interventions, such as the ability to tell people to wear masks or to stay socially distant from one another. Essentially non-pharmaceutical meaning non-vaccine related, non-viral load related intervention methods to help reduce or maximize the potential reduction of risk of infections. So here's a brief view of the gateway that we created in order to try and solve some of these problems, to provide further access and easy access to people that want to use the model and interact with the model. The link that you can see here will take you directly to this landing page for which there's a login system. And once the user manages to sign up with us, they're able to run their own computations and their own local models of what I've shown you previously on their own machine, using the supercomputer center at UCSD and using an allocation from Exceed, I believe. In the gateway, the different parameters that can be explored by the users for several different types of simulations, one for bus travel, one for classrooms, one for an entire school system, like you saw previously. And within these, you can explore the parameters you can see on screen, such as wearing masks, the different occupancy levels, testing rates, vaccination, et cetera, et cetera. Use of the gateway in each one of these simulations, like I just talked about previously. As you can see here, this is the bus simulation, where the image that you see on the left is the inputs for the user and what they see initially. And then once they hit run, after calculating for a few minutes, it produces several different plotted outputs, which are at the top, risk heat map to determine the distribution of the viral load approximately by the end of the trip, the likelihood density estimate of the probability of an average student being infected on a simulated trip, and then a seating plot to visualize how far apart the students are sitting with this capacity. And so as a result, we can see that the gateway makes it much, much easier for people to interact with the model and see both the input and output. The results, to explain a little bit closer, are seen here. The comparisons that are available are the ability to run multiple different simulations with different initial conditions, such as having 56 students on the bus, all of them wearing masks, and the trip is for 30 minutes. But one of the simulations are done, half of the simulations are done with the windows open and half of them with the windows closed. And as a result, the average rate of infection of the different students in the different seating positions, as you can see here, is on average lower for the simulations where the windows were open because of the larger ACH. With the result of doing significant amounts of simulations allows for the law of large numbers to show us the probability distribution of a lot of these simulations, even though we're using random initial conditions and random setup for a lot of the situations, which makes the model a lot more, it makes it a lot more consistent, but it does require a lot more computational power, which is another thing the gateway is able to handle. To just briefly go over it, this is another image of some of the results. This is from the school simulation, and it shows the results of initializing testing at different times, at the end of 1 week or at the end of 2. Ongoing work in the project is in several different areas. We've recently got a new batch worth of volunteers to help out, and several other students that are continuing in work. To do this, we've got the parameter sheet that is updating the table and supporting the model system is going from being hard-coded and supported by the parameter sheet to having API requests call from the various websites which we've gathered these resources and automatically update them when those resources update to keep this model up to date and to avoid a significant amount of future work to keep the model valid. Speaking of validation, the validation with the San Diego County of Education and San Diego Unified School District have also been things that we've been keeping an eye on, especially as students go back to school and we are able to gather data from the source essentially and see and ask for specific variables and such going in to see what they can track. We're also implementing variants in vaccines. We're updating our aerosol dispersal model to more closely fit to the research. We have more simulations in the works, such as some for sports activities. We've got a nurse dashboard and decision trees that are working towards the validation with the school district and with the County of Education, the former of which is intended so that nurses at these schools can take a look at the students from their school district and determine programmatically using these decision trees that you see on the right whether or not the student can come to school today, whether they need to be tested, et cetera, et cetera. And so on. We can also, we're also working on several different types of performance improvements, such as porting some of the essential critical code over to Julia to improve the speed and to parallelize a lot of the computation initially. And also we're going through and formatting the initial code to speed up how it's written in terms of the code itself. We have several acknowledgements. We've got thank you very much to SGCI for help with Theravada and Exceed. We've got thank you to the San Diego Health and Human Services Agency for the data, the school floor plans and support, to San Diego Unified for letting us take a look at their buses and for showing us the bus routes and their configurations, for the UCSD School of Medicine, in particular Professor Howard Taras for exceptional guidance, for the San Diego County Office of Education for the help with the nurse dashboard and for the decision tree, and for the NSF award for the NSF support for award 2139740. I believe that's all, and we have several different pieces of information in the appendix, which I didn't want to fit into the rest of it because it took a while, and I believe that's it. So thank you for listening to me, and thank you for bearing with me while I catch up. Are there any questions? I'm just going to open the chat now. I've been leaving it off to the side.
Yeah, that's a good idea. Good timing too, Bailey. An interesting talk. So we have 1 minute for maybe just one question.
I'll be around for the rest of the day if people have others as well.
Okay. Please put your questions in the chat box. I think Bailey will be looking at it.
Yeah, I'll go through now. Thank you so much.
Yeah, Bailey, if you could stop sharing. We'll go to the next paper. The presenter is Pajot. Pajot, could you take over and start sharing your screen?
Yep, great, thanks so much. Let me just make sure I do it in the right order here. All right, is that coming through okay?
Yes.
Okay, great, thanks so much. Yeah, so I'm super excited to be here. My name is Pajot Van Kay, and I'm here on behalf of the National Microbiome Data Collaborative team. We're quite a large team, and I'm representing over, I think, 70 individuals who are working really hard on this project, and excited to share some of the work that we've been doing, especially with respect to usability. So just a quick overview. Oops, there we go. Just a quick overview of what we are and what we're trying to do here. We are a pilot project funded by the Department of Energy based in the United States. And what we're essentially trying to do is making sure that multi-omic data, as you can see on the right-hand side here, the different types of experimental data that are generated by microbiome data are accessible and available to researchers. So currently these different types of omic data exist in various different primary repositories, as you can see on the right-hand side. These are some of the really popular ones over at the JGI, Joint Genome Institute based at the Berkeley Lab, as well as other ones that you might be familiar with, such as NCBI and EBI. And what we're trying to do is make sure that the metadata that describes this experimental data is standardized in a way such that researchers can actually access this information, search for it, and then actually seamlessly gain access to the experimental data. So one thing I want to clarify here is that we're not replicating the primary repositories, we're not replicating any of that data, but instead trying to make that data findable, accessible, interoperable, and usable. You can take a look at all of our GitHub links at the bottom here if you want to explore more about what we've been working on, but these are all currently in development, and our activities are going to continue to evolve as the time goes on. So really quickly here, metadata standards really are at the core of what we're doing here. So when we say metadata, some of the researchers that are out there might cringe a little bit because it kind of depends on what you're working on when you refer to metadata. So of course, along the research workflow, there's sample metadata, preparation metadata, of course, the processing metadata, and when you run your bioinformatic workflows and any of the annotations that get generated as a result of that. Most of what I'll be talking about in this presentation is focused on the sample metadata. So this is metadata that describes the environmental context in which you collected your biosample from. One of the main activities that our team is also working on is linking together the data standards that have already been developed by the community and making sure that those standards are interoperable, as well as facilitating conversations with the community so that we can provide feedback to the organizations who are developing the standards and improving access to these standards so that they're actually being used by the research community. One of the other main activities we're working on is making sure that standardized bioinformatic workflows are available for the community. And so, of course, as many of you may already know, that having standardized workflows really do enable cross-study comparisons. And so we have workflows available for each one of these omic types. And we're making them available in containers as well as through this GUI, which I've included a link on the bottom right-hand side here. We have a various number of metagenomics workflows available through our NMDC Edge platform. And users can just go ahead and go there and upload their data and just with a click of a button, run the workflow, and then be able to compare some of that data with what we've been generating through our NMDC portal. We're adding support for these other workflows. We're currently adding workflows as we speak here and looking for beta testers, and we're working really closely with the community to improve these workflows as well. But the main thing that I'll be talking about today is actually our NMDC portal. And so I'll be kind of demoing that in a second here. But again, just reiterating the fact that, you know, we're not a primary repository, but we are a repository for metadata. So of course, that refers to the sample metadata that I referenced to before, but then also the processing metadata, as well as, you know, the different types of metadata that I showed in that figure before. While this is a figure that kind of shows who we're already linking out to. So for example, on the bottom left-hand side here, our Department of Energy Resources, we're currently, you know, hoping to move forward with resources at NSF, as well as future efforts to hopefully link to NIH resources as well. So making sure that all of that data is accessible. So I'm just going to actually go ahead and do a quick demo of our data portal so you could see what I'm talking about. And this link is certainly available for anyone to go to and start exploring there as well. If you work with microbiome data at all, we'd love to, you know, work with you. So please reach out. So one of the main things that you could see here is that, you know, we have the different kinds of omic data that are available, and you can, you know, all of these little visuals are clickable, and you can refine your search based on that. We also have this text-based search that updates as you type. So for example, if you're interested in samples collected in Colorado, you could apply that search term. If you're interested in, let's say, soil samples, it returns a bunch of different search facets that have the term soil in it. So I can go ahead and click that as well. And it just further refines this interface so that you can find what you're looking for. Now, if I scroll down a little bit here, you could see that, you know, 48 samples happen to, you know, hit this search filter. And there are links out to the primary repositories here. So for example, this actually links out to the raw data over at NCBI, and it'll redirect you there if you're interested in that data. But of course, we have all of the data products that were generated by our standardized workflows that I mentioned before that the users can actually download individually or make use of our bulk download here. So actually download all of the data associated with the 48 samples. Also really quickly, I wanted to share that, let me clear that really quick, okay. Wanted to share that we also have this, what we're calling our upset plot. Essentially, you know, a lot of researchers have come in and wanted to find biosamples that have had multiple types of omic data characterizing that biosample. So for example, if I wanted to click on this 43 here, it'll return and filter by these biosamples. These 43 biosamples have had both metagenomic, metatranscriptome, as well as organic matter characterizations associated with them. So it's a really great way of searching for that across the board and then going and downloading the data that you're interested there. So I'm going to stop here for the demo and get back to my slides. So 1 second. Okay, And I also really wanted to share today some of the work that we're doing in terms of engaging with our community. And so throughout the development cycle of, planning, designing, and, prototyping, reviewing, and adapting everything, we are working very closely with the microbiome research community to make sure that what we're building is meeting their needs. So this process looks a bit more like this, where we're doing a lot of iterative feedback and deployment. So at each iteration since our release in earlier this year, our portal has continued to evolve. So I put a little screenshot here of what it looked like originally and what it looks like now. And it will continue to evolve with some time. So the next time you look at it, it's probably going to change quite a bit. And one of the really great things about this is that it's enabled us to ensure that the features that we're adding are relevant and useful to researchers, but it also allows us to improve access to the data that we're trying to make available. We really are borrowing from a lot of the software practices of Agile and Lean. I really love this figure in that, it's allowing for iterative development so that there's a minimum viable product at every stage, and not necessarily developing something like the above figure where you might not necessarily be able to get useful feedback from your user community at each stage. So it's been really great to work with them. And what this iterative feedback process looks like is that typically a request will come in, and we'll spend some time with several of our usability experts to develop a script and then identify interviewees based on our community and sort of who we're currently working with, as well as doing an open call there. And then we'll conduct the interviews and do a huge analysis of the interviews to pull out some of the insights that we've gathered from it. So, and then we'll take those insights and actually sit down with the development team to brainstorm, how do we address some of the issues or the insights that have come up. So really excited to share some of the features that were directly informed by these usability interviews. So for example, that global text search that I showed earlier was something that had come up repeatedly in our user interviews. It was just a little text box that all of our users wanted to keep typing in and expected to see certain things pop up. So that was a new feature that was added recently. Being able to pan, zoom, and search for biosamples geographically was also something that had come up there during our user interviews repeatedly as well. So we've added support for that. And if you play around with it on our portal, you can actually see how you can apply searches by clicking interactively and clicking on the button. And then this upset plot that I had referenced earlier, researchers had let us know that they were really interested in, sometimes only specific types of omic data, but they may want all of those omic types of data to come from the same biosamples, because they might want to, do some comparisons across that, and to make sure that, for example, maybe some, genetic markers are coming up across the board. And so that's really what inspired our motivation for creating this plot and allowing it to be interactive so that researchers can search and filter by that. And then last but not least, the ability to download data more than just one file at a time was certainly something that also came up during our interview. So we had very recently added a support for bulk data downloads as well. So we're really excited that this process has worked very well for us. We really love working with our research community, and with each of our quarterly releases, we also aim to do a round of usability interviews with each one of them. So that's essentially all I have for today. And I just wanted to reshare our mission, as well as links out to our portal. We do work very closely with our community on more than just the data portal, but on various other aspects. So I encourage you to check out our website and follow us on Twitter, and reach out if you have any questions. We also have a couple of publications that you could check out if you're interested, but happy to take any questions.
Thank you. Very nice presentation of a complex system design, software engineering, and with a live demo. Any questions from audience?
Yes, I would like to just bring to your attention the work that we've been doing for 15 years now, since 2006, on the Portal Doors project with metadata management. And we have recently renamed our Portal-Doors project principles the DREAM principles, where each one of the 5 letters, DREAM, stands for a fundamentally important principle, one of which was completely omitted by Wilkinson et al., which is how and why they were able to paraphrase our work without citing any of it. So I encourage you, if you wish to be fair, in the ordinary English meaning of the word fair, take a look at the Portal-Doors project, the Portal-Doors principles, and the manner in which they have been renamed as the DREAM principles. I will be giving a presentation tomorrow as one of the work sessions, and I encourage you to attend and take a look at our papers. They go all the way back to 2006.
Thanks
Great, thank you so
comment.
much for that.
Yeah. Okay, if you could stop sharing, we'll move on to the next speaker. William, please take over. Oh, sorry. Our next talk, sorry, William. Our next talk is from Matthew Cassandra. He will present a science gateway generated by GenApp. Matthew? Here we go.
Are you able to see my screen and hear me?
Yes.
Okay, great. So it's a pleasure to be presenting here today. My name is Matthew Casertano, and I'm a senior at Montgomery Blair High School. And I'm presenting on behalf of the 3 colleagues below who I've worked on this project with. And our project is NMR Suite, a GenApp generated science gateway for NMR data analysis and determination of structural ensembles of biological macromolecules. So the importance of the problem is that structural analysis of proteins and nucleic acids is extremely critical in biomedical research, and learning more about it can help us better understand the biological function of macromolecules as well as improve treatments for diseases. So biological systems formed by macromolecules like proteins are dynamic systems that move and adjust according to environment, function, binding, etc. And scientists have been interested in understanding these processes and ultimately understanding how a dynamic system works for a long time now. So in a single domain system, the focus is on dynamics of specific groups of a protein like side chains or the backbone itself. And so this is a challenge that has been studied with techniques like NMR, in addition to X-ray crystallography, structure determination. And although these techniques are well developed, most systems of proteins are multi-domain, which introduces another layer of complexity. And so for multi-domain proteins, it is a typical optimization under uncertainty problem because the experimental data are a convoluted average of contributions for multiple conformations. We're talking about proteins which inhabit multiple states of conformations, and studying these different conformations is very important in all kinds and types of systems of biochemistry because it's the key to understanding how biological processes happen in nature. And so NMR Suite offers tools for this type of analysis as well as for single domain analysis. And I will explain more about what these concepts mean in the coming slides. But first, before we get more into NMR Suite, let's take a step back and look at the framework which made this possible. That is GenApp, which allows researchers to rapidly build applications like science gateways. It helps to minimize dark code, which is code that is used in a limited domain only. For instance, a certain lab has it, but only on their own computers. And so the Fushman lab had programs to perform these types of protein analysis, but the programs can only be shared by direct download. So people had to have MATLAB, Java, etc. already installed on their computers. Plus every time the programs were updated, every person who had downloaded them would have to re-download. Plus the programs were not integrated and were all standalone. And there were also algorithmic inefficiencies stemming from the fact that they were not integrated. So this was the rationale for creating NMR Suite, which unifies all of these methods of protein analysis under a single roof. And this was made possible with GenApp, which brings projects to life. So an example of GenApp's capability can be seen in the Predict module. Predict is one of the modules that is in the NMR Suite. It enables ab initio prediction of the relevant conformers for the experimental data, covering the available conformational space for a given molecule. And so we can see on the top, there are many inputs that go into the Predict module. Many of these inputs are shared between the three sub-modules, predict PRE, predict PCS, and predict RDC, which essentially approach the problem from different angles. And so we recognize that there is some inefficiency here, and it would be much simpler and it would make more sense if these could all be combined under a single module, so that we would not have to run the same files repetitively from different programs. And so this was why Predict was one of the modules that were encompassed in NMR Suite. We will see what it turned into with NMR Suite. But first, the way that GenApp works is that it assembles applications from logically organized definition files. And we can see 4 steps, which is pretty much all that's needed to put a file on GenApp. So now the Predict module became this on the right. We can see we pretty much are able to perform all 3 types of predict analyses at once. We can simply upload the data we need. So all the shared variables which are highlighted, we only have to upload them once. We can run multiple types of analysis. And overall, it just streamlines the process and makes it much more simple for researchers. So another aspect of the NMR Suite is PAR-NMR, which is essentially approaching the single domain protein problem that I was discussing earlier. Sorry, that's my school announcements.
Will be Misa makeup. You will
But
receive an email
so NMR
and or a
Suite.
connected message
Okay,
stating
I guess
you
I'll
are
wait
eligible
for.
to participate in Misa retake.
Other audience, please mute.
Check your student mcpsmd.net account for this testing invitation.
Matthew, please go ahead.
No, that were his school announcements. So that
Okay, sorry. Yeah, my announcements are over now. So the NMR Suite contains a set of modules which perform analysis of experimental paramagnetic NMR data. This is essentially single snapshot data of a protein in a single conformation, or at least treating it as if it inhabits a single conformation. And so there are 3 submodules here. Each one of them takes in each one of them outputs different information about the protein. And these 3 are all implemented within NMR Suite. Then we get to conformational ensemble analysis, which is that multi-domain protein problem. The goal of this is to uncover the conformational ensemble of proteins based on underdetermined structural data. And so the first step is using the predict module, which was mentioned earlier, to generate an ab initio prediction of the relevant experimental NMR data. Then we have 2 algorithms that come into play. So essentially, we're generating a bunch of structures that the protein could possibly inhabit. And the question is to determine which one of them are relevant and which ones are not. And also for the ones that are relevant, what weight we are assigning to them. So these are essentially the 2 questions that we're asking about: what conformations best represent the ensemble based on the experimental data, and then what are the relative weights of each of these conformations to determine what states the protein is inhabiting. So for deriving this solution, we can start with an unconstrained problem, which is simply trying to minimize the difference between the experimental data and what our hypothetical model would predict the experimental data to be. This is an ill-posed problem and has infinite solutions, and it's unstable. So we can regularize it by adding a regularization parameter that's lambda times L of x. And this becomes a well-posed problem. It recognizes the fact that we're dealing with limited experimental data, and it has relatively few solutions. And we'll see 2 possibilities for the regularization parameter that are used in NMR Suite. The first one is max ent. This runs a maximum entropy solution. So it's multiplying each weight by the log of itself, and again, multiplying that by the lambda regularization parameter. And so what this essentially does is it essentially tries to make the least number of assumptions possible about the experimental data. So it's just a typical maximum entropy approach. And we can see the L curve on the right, which shows as an entropy increases, we get a better solution, but eventually there's a cutoff point where the improvement is slight. And then there's also SES, which stands for sparse ensemble analysis. In this case, our regularization parameter is simply the number of non-zero weights. We're looking to minimize the number of conformations that we assign a non-zero weight to, essentially asking what is a minimum number of conformations that are required to explain this protein. We can see the L curve plot here. It shows that as the ensemble size increases, the relative error also decreases, but at a certain point, the trade-off or the benefit becomes minimal, and at this point, the program stops. So summary is that NMR Suite is a science gateway powered by GenApp with tools for protein modeling and analysis for both single and multi-domain structures. It is being used as a teaching tool in biochemistry classes and to assist with groundbreaking research. It combines previously standalone modules under a single roof. It can be found at the following link. And this will hopefully encourage other researchers to also share their software in a similar way. We'd like to acknowledge our funding sources and the many people who contributed to this project. In particular, I'd like to thank SGCI for funding a large portion of my internship work. Thank you everyone for listening, and does anyone have any questions?
Thank you,
I
Matthew.
apologize for the interruption.
No problem. Nice presentation.
Thank you.
Any questions? Any questions from the audience? So I have the one. Is Gen app specifically for this bio app, or can be generalized to support any kind of app codes and integrated into this
Yeah, so that's a great question. So it's something that can be generalized. So for example, on this slide, we can see like an example energy calculator. On the left, the parameters are defined for the interface, and on the right, the interface is produced. So it's really something that can be generalized to any type of application. It's meant, just like as a general way of producing scientific gateways for researchers.
And the application you generated, is it a web app, desktop
Yes.
app, or mobile app?
It's a web app.
Okay.
Yeah. Yeah.
Thanks.
Yeah, of course. Great question.
Thank you for the nice presentation. I
Mhm.
hope this project will motivate you more for the next phase development. And in the interest of time, let's move on to the next talk. Matthew, if you could stop sharing. Our next talk is from William for the platform for epigenomic research. William, okay, good.
Do you see my screen all right?
Yes.
All right, thank you. Thanks for having me. Been some great talks so far. I want to talk to you about a platform that's been in development now for several years with my group that we built to handle the epigenomic and genomic research that we produce a significant amount of. So I am not a computer scientist. I am actually a biologist, and this is the question that kind of underlies all of my research, which is, how do proteins actually organize themselves on their genome? I'm not going to get into any of the biology of what we've done, other than say that I spend a significant amount of my time every day mapping where all these proteins actually bind in a variety of different organisms, ranging from yeast all the way up to human. And when I say generate a lot of data, what I mean is that in the last year now, we have published 2 papers that constitute 3,000 different genomic data sets. And anyone who does this level of genomic research knows that while we published 3,000 data sets, the actual amount of data we generated was probably an order of magnitude larger than that. So I'm talking probably somewhere in the order of 30,000 data sets over the course of these projects were generated just to get the publication level data that we needed to get these papers out the door. So this is an enormous problem in terms of management. Not just storing the raw sequencing data, also storing the process data, and storing the data, organizing it, and then linking the individual sample meta information to that data. And the final problem, which really doesn't have a lot of good solutions, although there are many options out there, is once you have that data, it's organized, it's stored, how do you get it out to the larger community? And kind of underlying this enormous question right now is the growing pushes from both the NIH and the NSF for we need better rigor, reproducibility, and the ability to disseminate all of our data. So when we started these projects, which started many years ago, we were faced organizationally with the fact that this is the gold standard for organizing an academic laboratory, which is after several decades of the group I joined working, we had literal piles of lab notebooks that digging through them would reveal what the actual experiment was that was on the actual server somewhere. And there was no information other than that. So we started with the question of, okay, let's break this into kind of two different main problems. We have biochemical reproducibility issues and we have bioinformatic reproducibility issues, because also up until, about 10 years ago, all of the bioinformaticians in our group were making, one-off shell scripts that disappeared into the ether once they left to move on to a different group. So luckily for us, bioinformatic reproducibility is handled very beautifully by the actual Galaxy Consortium project. For those of you who are not familiar with this, Galaxy is a web-based bioinformatics system designed for 100% reproducibility. We were lucky that when we started our project, we were located at Penn State at the time, and Anton's group, which is where the Galaxy main team is based out of, was located, the building next door to us. So his group was awesome and instrumental in terms of getting our own custom Galaxy system online. And the beautiful thing about Galaxy is that when you load all of your bioinformatic tools in there, it encompasses everything for eternity with perfect reproducibility. And it actually lets you in a very nice GUI interface actually chain different tools together so that when you set off a workflow with certain data inputs, it will automatically go from start to finish to the very end, and it will track every single aspect of every single tool that ever runs, not just the tool parameters, but also the specific CPU it ran on, the version of GCC compiler, everything with timestamps for everything. And this is stored in perpetuity on the Galaxy system. So this already existed when we started our project. So we felt that it was in our best interest to actually adopt this for our bioinformatics. So, although bioinformatic reproducibility in our projects was, considered solved on our end, we had this larger question of, well, how do we solve biochemical reproducibility? How do we take everything that we learned when we spun up our own Galaxy instance and applied it into biochemical genomic research? So we ended up developing this platform for epigenomic research, spearheaded by a fantastic software engineer, Danielle. And the idea was, can we make an open source meta information tracking system that kind of mirrors this Galaxy system, that allows you to have an idea of an experiment, match it to versioned protocols, versioned inventory, do the experiment, do the sequencing, do the bioinformatics, make a discovery, and finish with a publication. And basically the goal was that we set out was that the publication figure that you click on when you go to the actual paper, we should be able to track every single aspect of every single material, computer resource, script, parameter, at biochemical wet bench protocol, all the way back to the beginning that generated that figure. That was the vision we set out for. And year after year, we get closer and closer to that. And again, kind of mirroring the ideas of what did we learn from the Galaxy system, we tried to mimic as much as we could from what we learned bioinformatically into this biochemical aspect. So we included things like a QR barcode scanner that our technicians use, so that when they're actually physically doing an experiment, they can scan the reagent and it will automatically populate the meta information system that is linked to the experiment and the protocol they're doing. So the larger vision is that we can replace lab notebooks with a tracked meta information system that will allow us to, on any computer, because this is a web-based application, that we can go back in time and see what any given member of the lab did on any given day with a complete record forward through backwards of what they did. And then because we are very heavily invested into the Galaxy system, the idea is that, well, then we natively integrate with our Galaxy platform with RESTful API calls, and all of the bioinformatics that Galaxy is performing, we will actually be able to integrate directly from a visualization standpoint into Pegger, so that we're able to look at quality control metrics as we go throughout the experiment design reiteration process. So the way we did this was essentially every single row you're seeing here is a different sample that's going through a different Galaxy workflow instance. And if we go to that workflow, what we're actually doing is, I showed you that GUI before, where this goes from tool to tool to tool doing an analysis. At various points of interest for us, we actually have a companion tool that will send a post request to our Pegger system containing all the meta information that we want to track related to this. So what this actually does from a practical standpoint is that any given one of these samples, we now actually have not only the Galaxy history ID that links us to the complete processed workflow. For each individual sample now, we're able to actually have a unique ID code for every analysis step that was ever performed on the sample. And one final thing is that, due to, issues with data sets failing due to the experiment failing or, the server failing, we're also able to encode success or failure messages here into our output statistic messages so that users will, as they use it, are able to know whether or not, oh, the HPC system is down right now, or your experiment actually failed and we're stopping the workflow here. So in order to make this come alive and make it more practical, Pegger includes a project management standpoint or system, which lets you essentially have different projects that are user-controlled version or with admin capabilities and all that jazz going with it. Allows you to assign different samples to it. Administrators obviously have the highest amount of power in terms of assigning it, but we also designed this so that we could have guest accounts that we could send to our collaborators so that they could view only the projects assigned to them for our collaborative projects. This includes a comprehensive inventory management system that not only tracks every single bit of inventory in the lab, but also versions it and assigns it a unique QR barcode that with our web or with our mobile front-end app, we're able to link for any downstream project. And then we have our sequencing run instance. So most people multiplex samples when they run sequencers. So it could actually contain samples from a variety of different projects. So by leveraging all of this together, we are able to, for any given sample, we can link it to a specific sequencing run, to the inventory it was using, to the protocols it was using, and et cetera, et cetera. And all the way down through, these are dynamically generated reports that are actually literally pulling data live from our Galaxy instance to visualize the specific workflow meta information that we want. And the goal is to actually make these reports more and more generalizable over time and to support a growing number of genomic assays. So at this point, the current status of our Pegger system, we've now recently basically expanded this now to support pretty much most common single sign-on options, including both SHIB and web access. I always say we always code for the least common denominator. The goal, in terms of a science gateway is that we also understand from the idea that we're, looking to also begin doing more patient clinical stuff, that these things need to be lightweight and able to run on an individual, server that may be air gapped off from other networks with regards to patient medical information. So we always program to be ultra lightweight. So our production Pegger that supports our entire group runs on a single CPU core with 8 GB of RAM, and its complete footprint that encompasses about 40,000 different sequencing samples across 500 runs is less than 100 gigabytes, of which most of that is actually the PNG gel images that users upload to assign to their different samples. If anyone's interested in this, we have the manuscript currently up on bioRxiv right now. It's under review, so if any reviewers are here, please have mercy. And our code is open source and available for download. And we're always looking for people to collaborate with, to streamline this, to expand it to not just how we use it, but we really think that this is a true end-to-end solution for solving a lot of the rigor and reproducibility issues that exist in the biochemical field right now on the genomic side. So with that, I want to thank the many, many people that have contributed into this project over the year. Specific shout out to Danielle Shao, who's still at Penn State, who did an astronomical amount of work in terms of making this happen and getting our preliminary prototype systems off the ground. So with that, I'd like to take any questions you guys might have.
Any questions from the audience?
If I may. So I noticed that you used the sequence logos of Tom Schneider, and I'm sure Tom will be pleased. But one of the issues that has come up, that's an example of a methodology, a technology, a device, whatever you want to call it in terms of software. It's a technique. In the current era of not bothering to cite, when is something like that, when should it be cited, when should it not be cited?
So and maybe if I could just say the question again, are you asking when would one cite this software package?
It's a general question about one of the current problems in the internet web data sciences era of when a data set does or does not get cited, when a data technology does or does not get cited, when a paper involving either a software technology or a software technique, a data set or data repository, et cetera, when should or should not items, regardless of what kind of item it is, be cited or not cited. And Tom Schneider's sequence locos is a perfect example. I know for a fact that many authors use his sequence logos without citing them, without citing him as the original paper for the device of those sequence logos. So when should things be cited or not cited?
Yeah, so that's a great question. I think that's something that a lot of people who run science gateways and also core facility directors struggle with, which is if you're providing a service, when and how do you get cited? So, I can tell you our personal game plan is that upon publication of our software platform, we are getting it registered to have an RRID, and we will be acknowledging that RRID in all of our publications going forward to reference our utilization of the software. And we can't force anyone to acknowledge our software if they use it, but we would hope that if they find value in it, they would certainly cite it so that we can continue to get funding to, further development on
it. If I may just lend my voice, I am quote unquote old fashioned, so I always do believe in citing correctly and properly. And so I would encourage any who use any kind of item, whatever kind of software it is, whether it's a data set, a data repository, a software method, a software technique, a software technology, that the original paper or original author should in fact be cited and credited. So, I happen to believe the sequence logo should be cited to Tom Schneider. So that's my opinion.
Well, it's a good topic. We can continue in the next social interaction session. In the interest of time, let's go to our last talk. The presenter is Ali. He will present Stencil, a web templating engine. Ali?
Yes, thank you. Yeah. Can you see my screen?
Yes.
Okay. Yeah, I'm going to talk about Stencil, a web templating engine for visualizing and sharing life science data sets. My name is Ali Nematbakhsh. I'm from Epigenomics Core at Biotechnology Institute at Cornell University. First, let's talk about the importance of the web-based data visualization platforms. In this graph, on the x-axis, you see years from 2009 to 2021, and the y-axis, you see the amount of data that is registered now in the sequence read archive. And you can see the amount of data reaches to about 10,000 terabytes of data. And it's very easy to get lost in this amount of data and how much information we can get out of it. One obvious solution is, of course, we can get graphs and plots to get more information and inference out of the data. These graphs, however, works better if we can have interactive plots. So we can zoom in the special place that we are more interested in, or we can like see more details of the specific location in the plot. So interactive plots could enhance our understanding of the data. Also, if it can be the data or the plots can be and graphs can be shared through the different devices and become a web application, it gets it easier to be shared and be spread among the users, and it can benefit the society. In light for that, there are lots of popular web-based interactive data visualization platforms has been developed. One of them is RShiny, which is trying to make R packages available to through the user web interface, and lots of bioinformatic tools that has been developed through the web interface by using RShiny, such as ChIP-seq and RNA-seq, two very popular assays in bioinformatics. Another one, which you might probably know, is -Python. It's an effort in the Python to give web access to the Python codes and in a easy platforms. Stencil is trying to help with ease of, in this direction, it's trying to help not only to give the web accessibility, it also wants to help in reproducibility of the generated plots. And also it's an effort to also support the codes that are coming from different languages. So Stencil software, this is a architecture of that. It basically uses more a classical way of having a front end and a back end, and also a database. All it needs is it needs some JSON dictionary to be posted to the database, and this JSON dictionary can come from custom tools, Python code, a Java code, or whatever, or it can come from the workflows in Galaxy, which will in the previous presentation talks about it. I'm also going to give a slide and talk about it in a slide. After that, it goes to the back end and front end. Then we can take advantage of lots of interactive libraries that are developed in JavaScript and can have interactive plots. So based on that, then we can generate different interactive plots that a user can interact with it. And these are, for example, based on the Nivo plot, which is on top of D3, which is a library in JavaScript. So basically, we are taking advantage of already developed JavaScripts. Talking about integration with Galaxy, let's just give you a short introduction about Galaxy. Galaxy is a web interface initially developed for bioinformatic analysis and now gets popularity in other fields as well. It does a very good job. Made my job easier. So I just say it does very good job in tracking metadata information and making sure all the results are reproducible. So it tracks all the metadata that has been used for getting a results or plot in our case. Also, it does a good job in high throughput computing environments where lots of data needs to go through the same workflow. So it can handle this kind of environment. What it is, it has a web interface. It has on right-hand side, you can see history, all the steps that is used to generate a plot and the workflows that is used for generating that plot. And on the left-hand side, you can see the tools that you want to use. In order to integrate our stencil with Galaxy, we develop the workflows, and then, for example, this is DSeq2, a very popular tool in RNA-seq analysis, and then we develop some pre-processing script, which change the data to a format, JSON dictionary format that can be interpreted by our stencil project. And then we have a simple post function that posts the results, the data for that plot or table to stencil. After that, the results are ready to be shown. So this is a live example. This is an example of a website that has been developed by our lab as an example of using stencil. So it has a pie chart here, which is an interactive chart, and also this website is for showing how the proteins are interact with DNA. You see this pie chart is interactive, so when you scroll around it, you can see the data. It shows different tissues and cell line that has been used. For example, you choose a protein, and this protein, after you select, After you select the protein, it goes to the to that information about that protein. You can see some static plots here, a dynamic plot that you can click on it and get further information out of it. You can close it out and have a download button to download the data. Also, you have another dynamic plot, some bar charts, which you can use. You can interact with it, some other bar charts, and also you have another line plot, which is you can see it's dynamically changing as you click the tabs. So basically, this is an example of this using stencil in an application. Finally, in conclusion, I was to just say that we developed Stencil as an interactive visualization platform and integrated with Galaxy software to generate trackable and reproducible plots in high throughput computing environment, which we hope that it can be useful for our community as well. At the end, I would like to thank Chi-san and from BioHPC and Prashant Kontala, former member of the lab, for developing the front end and back end of the software. Greta Kellogg, Frank Pugh, and William Lai for supervision, and other members of the Pew lab for their support. And if you have any questions, please let me know.
Any questions from the audience? Very nice talk. So let's thank our speakers with a virtual applause.